So bicornuate uterus is actually pretty high yield for the step 1. They really want you to know that it's due to incomplete fusion of the paramesonephric ducts. Okay, incomplete fusion of the paramesonephric ducts. So when we have fusion of the paramesonephric ducts, we get a properly formed uterus. Okay, but it's incomplete fusion that leads to the bicornuate uterus. Now, why am I pointing this out so explicitly? Is because USMLE will classically show you a picture just like this one, and then they'll have as answer choices the mechanism for what caused it, and you'll see the image, you'll say, okay, well, that's a bicornuate uterus. And then choice B might say incomplete fusion of paramesonephric ducts, and then choice C might say complete failure of fusion of paramesonephric ducts. Okay, so if we had a complete failure of fusion of the paramesonephric ducts, we'd actually have a double uterus, double vagina. Okay, but it's an incomplete fusion that leads to a bicornuate uterus. So remember the incomplete, not complete. And this can lead to infertility and recurrent miscarriages. So the SRY gene on the Y chromosome produces testes determining factor, and this is what is very important in ultimately deciding the fate of development of the initially generic gonads, okay? Because our default will bring these generic gonads to become the female phenotype. But testes determining factor is what will lead to the production of the Sertoli and Leydig cells, and our Sertoli cells will secrete that Mullerian inhibitory factor that will cause the paramesonephric ducts to regress, and then our Leydig cells will secrete the testosterone that will be necessary to give us the male external phenotype through DHT and the internal phenotype of male through testosterone itself. So if we have a defective SRY gene on the Y chromosome, then we get a female phenotype because we wouldn't have Sertoli cells to cause the regression of the paramesonephric ducts, so we'd get the female internal structures, and we wouldn't have Leydig cells, so we wouldn't be able to produce testosterone to have a male phenotype. Okay, so without the SRY gene, we go down that default pathway to become a female phenotypically. So these would be men genotypically. Okay, they'd be 46XY, but they'd be female phenotypically because they don't have that SRY gene. Now, male versus female genital homologies, these are very important and they can be tricky. The USMLE will mention a structure on a male versus female, and then they'll want to know what the corresponding structure is in the opposite sex. So, in this case, the glans penis corresponds to the glans clitoris in male and female respectively, and they're both derived from the genital tubercle. We can get a condition called epispadias, which is when we have the urethral meatus on a male open on the dorsal or upper aspect of the penis, and that'll be failure of migration of the genital tubercle properly. That's one way they like to ask it. Or they'll just say, we have a glans clitoris, and what's the analogous structure in a male? It's glans penis. The genital tubercle also becomes the vestibular bulbs in a woman, and it can become the erectile tissue in a man. Okay, so this is the excitatory erectile tissue in a woman, and also the excitatory erectile tissue in a man. Okay, so we can get during REM sleep, for instance, or during any sexual excitement, arousal of this tissue derived from the genital tubercle. Now, the urogenital sinus, it will lead to the development of the greater vestibular glands. These are also known as the greater vestibular glands of Bartholin, and also the bulbourethral or Cowper's glands in a male. And similarly, we can get the urethral and paraurethral glands, okay, in a woman, as well as the prostate gland in a man. Okay, so those are from urogenital sinuses. And then the urogenital folds will lead to the labia minora in a woman and the ventral shaft of a penis in a man. Ventral shaft of the penis is the underside of the penis when it is erect. If we have the urethral meatus opening on the ventral aspect of the penis, that's hypospadias. Okay, USMLE loves to ask what caused the hypospadias, and it's failure of the urogenital folds to fuse properly. Okay, so you really need to know urogenital folds goes to hypospadias, whereas our genital tubercle will go to epispadias. Okay, so those are two pathologies on a man. USMLE really likes that. Okay, so as with our branchial cleft, arch, and pouch derivatives, it's heavy memory-intensive stuff. This is similar. I mean, a lot of this embryology is memory-intensive. But unfortunately, it's something you just have to look into before your exam. And then we have our scrotum, which is derived from the labioscrotal swelling, and that's the labia majora in women. Okay, so labia majora in women will correspond to the scrotum in a man, and our labia minora in a woman corresponds to the ventral shaft of penis in a man. Okay, so we have these respective structures. Now for the flash quiz, embryologically, the labia minora in a woman is analogous to what in a man? Exactly, as we just discussed, it's the ventral shaft of the penis, and they're both derived from the urogenital folds. Now, hypospadias and epispadias, okay, and as we just discussed, it's very high yield. Hypospadias is when your urethral meatus opens on the underside or the ventral aspect of the penis. Okay, so ventral / underside. Whereas our epispadias is on the dorsal, which is the top of the penis. Okay, so the part that faces the sky. And USMLE occasionally likes you to know that epispadias is associated with bladder exstrophy. Okay, so bladder pathologies. Okay, so bladder exstrophy is a common pathology associated with epispadias. Occasionally you'll see that in questions. So the descent of the testes and ovaries, there's something you need to know called the gubernaculum, which is this band of fibrous tissue that facilitates the descent of the testes in a male and the ovaries in a female. The testes and ovaries will start in the abdomen, descend inferiorly. The testes descend more inferiorly than the ovaries do. The testes will pass through the inguinal canal, but the gubernaculum helps anchor the testes to the scrotum, whereas the gubernaculum in a female will become the ovarian ligament and the round ligament of the uterus. The ovarian ligament attaching the medial pole of the ovary to the lateral uterus, and our round ligament is what attaches the fundus of the uterus to the labia majora. And then we have our processus vaginalis, which is an invagination of the peritoneum that will form the tunica vaginalis in a male, and it's obliterated in the female. And the highest yield thing you need to know, like practically the one thing that the USMLE will test you on regarding this, is that if this is patent, if you get a patent processus vaginalis, that is what causes a hydrocele to form. Okay, so that's when we get the transillumination positive. Okay, so light can pass through the hydrocele. It's not a cancer, but they'll ask the mechanism, and it'll be patent processus vaginalis. Okay. Which of the following is the adult male counterpart of the genital tubercle in the embryo? Okay, so it's the glans penis. Our bulbourethral glands, also known as Cowper's glands, those are derived from the urogenital sinus. Okay, so the bulbourethral glands are from urogenital sinus, and those are analogous to the greater vestibular glands of Bartholin in a woman. We have the glans penis, analogous to the glans clitoris in a woman, both derived from the genital tubercle. Okay, so glans penis is our answer here. We have the prostate gland, which is derived from the urogenital sinus, and recall that the analogous structures in the female are the urethral and paraurethral glands of Skene. We have the scrotum, which comes from the labioscrotal swelling. It's analogous to the labia majora in a woman. And the ventral shaft of a penis is derived from the urogenital folds, and that's analogous to the labia minora in a woman. To perform its unique function, the sperm cell has adapted many common cellular organelles into highly specialized structures. What is the function of the original structure from which the organelle that allows the sperm to penetrate the zona pellucida is derived? Okay, so the answer is vesicle-mediated transport, because what is the organelle involved here? It's the Golgi apparatus, okay? So it's the Golgi apparatus, and what's the structure that allows the zona pellucida to be penetrated by the sperm? It's the acrosome, okay? So the acrosome on the sperm releases enzymes that allow for the penetration of the zona pellucida, and this is derived from the Golgi apparatus, okay? So that's important to know. Our flagellum of the sperm is derived from microtubules. So if we were talking about flagellum, we'd be looking at cell movement, because that's microtubules. Okay. Cell-to-cell contact can be any of your junctional proteins. DNA transcription, RNA processing. Okay, so this would occur in the nucleus. We have oxidative phosphorylation, and that's part of aerobic metabolism, and that would occur in our mitochondrion. A woman gives birth to twin girls via cesarean section. The obstetrician notes that although each twin was contained in her own amniotic sac, there was just one placenta. What kind of twinning is described in this case? So it would be monozygotic twins that split after day 3. Recall that in this case, there's just one placenta, okay, so that's one chorion, so it's one implantation site. Each implantation site has one placenta, that's one chorion. But we have each twin in her own amniotic sac, so we have 2 amnions. So we got to look at this and say we have a monochorionic diamniotic pregnancy, and which of the following answer choices relates to that? Well, recall that if monozygotic twins split before day 3, we're going to have 2 separate implantation sites. So this would relate to 2 chorions, each with its own amnion, so dichorionic diamniotic. But if we get a split after day 3, between days 3 to roughly day 8, we'll get 1 implantation site, so we get 1 chorion, but there's enough time for the amniotic sacs to develop independently, so we get a monochorionic diamniotic pregnancy, which is our answer here. And dizygotic twins that split after day 3, this is actually not even true, because if you're a dizygotic twin, it's 2 separate eggs fertilized by 2 different sperms, so there's no splitting that occurs. And a conjoined twin pregnancy is when we have a splitting after days 12 to 13. Okay, so at that point, for conjoined, we have 1 chorion because there's only 1 implantation site, and we have 1 amnion, and we have a Siamese pregnancy or conjoined twin pregnancy.